and then I'll turn you guys loose to work on them and then ask questions. So to help you, given people struggled with the histology slides for respiratory, again, De Fiore is there. It's pretty good, but there are more drawings than they are actually pictures. But there is one photograph of the glomerulus and kind of drawing next to it that looks pretty much what your search image would look for. So to help you further, kind of like in the respiratory, a little belatedly, I posted some labeled pictures, and only one of them is the same picture as the ones in your catalog. That way, you'll still have to go searching for these things and know what to label. But you have some labeled versions. So I took down the old study guide. I'm going to be doing this periodically since I haven't finished doing like the whole digestive system in that study guide. So be aware that revised versions of that will show up. So the one I put up today has 2 new slides, and I thought I could share that new version with you so you can see. Well, maybe before we go there, why don't I first share the plaster nephron model. The other things in the nephron model, urinary models. Oops, urinary models. I thought I had this open. So I showed you the whole thing and stepping back, and I'm just going to, hopefully you're seeing this, because sometimes when I motor around here, are you seeing this? Somebody give me a thumbs up if you are. Oh, great. Thanks.
See you, Professor.
Yeah, so here is that rather battered, old, it's as old as the school, so it's probably 55 years old, something like that. You can see it's plaster, it's all cracked and chipped. But here are glomeruli where that filtration is going to occur. So
I'm sharing the slide.
you're not, I'm not sharing it?
Yeah, it's.
You don't see the picture?
Oh, no, yeah, we don't see the slide. I thought you meant something else.
Okay. You're seeing my desktop and not the slide. Okay, let me get back to, yeah, sometimes it seems to let me motor around and then other times it doesn't. Let me make it bigger now. Okay, so here are those green balls that filtration is going to occur in. So this is a pretty bare bones, my bird is going crazy now, bare bones model of the glomerulus, but I'm going to show you in the relief model that what it does, it zooms in and you can see in more detail the glomerulus. This one is particularly good for showing the blood vessels. So I said this is the arcuate this morning. These are the cortico-radiant. And then you'll soon learn this is the afferent arterial and this is an efferent arterial. So again, that shows up on the relief model. So in addition to that whole small picture of the whole model, I did some zooming in like this. So there's a whole bunch of these plaster ones. Okay, I'm going to stop sharing that unless you guys have a question and start sharing. I think my son is going to, not too many windows open. Where's the models again? Show you the other model. Maybe I'll have to go out of this one. Go back in. Okay. So let's start with this one. Let me turn it first. And keep turning it till I get it up right. Okay, now I'll share this one with you. So this is the newer version, the one I called relief model in the descriptions when you go to look at the pictures. So this one, it also has the arcuate. So you can see the medulla much more clearly. It looks more realistic. The cortex out here. And this is the only thing, one of the things I would argue with this model. Again, out of proportion, this should be much longer, right? But then they have the cortical nephron going well into the medulla. And so if I were to ask about a cortical nephron, I would probably use the other model, because this one is confusing. Since this is meant to be a cortical, this is juxtamedullary, but they show the cortical one go way too far into the medulla. So how do I know? Well, remember this was thin for quite a long segment, and then thick. And I would even argue with this that this is actually longer down into the medulla, not so high up, looking more like the pictures in your textbook or lab manual. So proportion-wise, there's some funniness. Here's the collecting duct. So those were the loops of Henle. And then in this case, they don't give a good representation of how elaborated the PCT is. So they made it look quite small, whereas the old model, the plastic one, it's more like what it is in real life. It's quite long. And then this is meant to be the DCT, which again is not very curly or elaborated. So the other one had the blood vessels here. If we zoomed in, which I could, but I don't think we need to at this point, you would see that there's an afferent, an efferent arteriole like before. But what they did, since the blood vessels lay on top of the nephron, is in this case, the other case, what they did is they had them lay on top, but they didn't show the full elaboration. They had like patches of blood vessels, and we can go back and look at that if you want. In this one, what they did is they just showed, well, if we only had blood vessels without doing any of the nephron structures, it would look like this. And then over here, you have the nephron without many blood vessels on top of it. When in fact, these blood vessels, which are going to be called the peritubular capillaries, they lay on top of both nephrons. And the same with this is called the vasa recta capillary bed. It lays on top of the loops of Henle. These that go nowhere don't make any sense, so just don't pay any attention to them. This is meant to be more like what the vasa recta look like. So over here are the interlobular, the arcuate, and the cortical radiant going up and down here. So a little bit different strategies between the two models. So I wanted to try and make sure you understood those. I also took, not as many, you could zoom in with this one a little bit better, but this model actually has 3 parts to it where they zoom in. So let me stop sharing this one. And again, you can't see it now, or, oh no, that was before. Okay, let me show you the frontal kidney. That's this one. Nope, that's not the one I was looking for. Oh, where is it? Oh, here it is. They show you the glomerulus. Oh, I got to turn it around first. It's rotated. Okay. The glomerulus in much better detail than the other one by kind of blowing it up and enlarging it. So we will be talking about this in lecture on Thursday, which will have to happen in lab. But here is the nephron, this is the glomerulus, and it's a tubule, so it has to have 2 layers. You can see the beginning of the PCT here with 2 layers and a lumen in the middle. Well, this doesn't look like it has 2 layers, but it does. This is the lumen, which is called the glomerular space. Glomerular, I know, I'm not remembering space, maybe. And then this is the inner layer. The inner layer actually coats the outside of the capillary bed. This is the glomerular capillaries. So on this right side, they got rid of the podocytes, the visceral layer of the glomerulus. And on this left side, they left the podocytes. So you can see the capillaries here that underlie that underlie these cells here. So we'll be talking about these podocytes that are a very specialized, simple squamous endothelial cell that enables filtration in much more detail. So if you look, this is the afferent arteriole, and you know that one because it's a lot bigger diameter, and that's really true in even more dramatically than this model. And the other reason is these cells on the outside, these are the granular cells that make the renin. So we're going to talk about the conjunction of, not every one of the members is shown in this model, but something called the JG apparatus, which is this portion of the DCT and its cells called the macula densa, the granular cells here, the afferent arteriole, efferent arteriole, and then not shown in this model, there are cells called mesangial cells, one population found in between here, between these capillary branches, and another population are found between this macula densa area and these blood vessels. So why I wanted to spend some time with the model is once we get to looking at the slides, everything is jammed together. And like the little green balls, all the glomeruli are these three-dimensional round balls that as the knife goes through them, you don't always get to see all these parts, these members. But if you get a perfectly, well, not perfectly, but if you get a nicely positioned section, you can see the macula densa here. You can often tell there are blood vessels. You can't tell which one is which. Bringing blood in, and I guess he's outside now. And you can see this parietal layer and the space here, the capsule of the glomerulus, where the filtrate will accumulate. So if you have questions about this, let's wait before we go and look at the slides that you're going to be drawing and see if anybody can put it in chat if you have some question about this. Okay, I'm not seeing any questions. Okay, so I stopped sharing that. Is there anything else I wanted to share? Yeah, so there's just the two pictures of that relief model, although I'm a little surprised because I had one of them shows the whole kidney. Maybe that's this one. Oh yeah, this is the third part of this one. Let me share this one with you. There are three parts to this model that we purchased. They were on different pieces of backing. So they're not always, oops, I didn't share the right thing, sorry. I can't talk and stop share that and start sharing the correct thing. This one is kind of what we talked about last time we got together, where you have a frontal section through the whole kidney. And this one shows you the blood vessels well. So the renal artery and the renal vein, the segmental, the interlobular, arcuate, cortico-radiant. And again, the little white balls are lots and lots of glomeruli. You know, the pyramids with where you will find the collecting duct and the loops of Henle, which they're showing here, and of a juxtamedullary nephron. And then, of course, the calyces, where as the urine comes out of the collecting duct, it is dropped into the calyces that go eventually into the ureter. So we did that part last time. Okay, so I'm going to go to the slides. I want to make sure you get some time to work since you're here and not everybody is. And that way, let's see, I had an order I was going to do them. Glomerulus with MD. It says, here it is. Okay. So let's start with this one. flying by me. Okay. So there are, in this view, 3 full glomeruli. That's these circular structures here. You are seeing this, right? If you're not, you can just mute yourself and say something. You see a little sliver of one over here. Great. Okay, so I focus this on these because you can get the sense that sometimes, like for example, this one, you can see there is a white space all around the circular structure. That is the capsules where the urine, the space between the 2 layers, the parietal and the visceral layers, where the filtrate, it's not really urine yet, is going to accumulate. So each of them have it. But some of them, you can see there's some structures and sometimes almost an indentation like that. The indentation is not as obvious on this guy, but I'm going to zoom in on that guy in a second because you do see the macula densa and the mesangial cells, and you see blood vessels, but you can't tell which is the afferent and which is the efferent very well. The other thing on this one you can see pretty well are these flat parietal cells that make up the wall of the capsule. So we'll zoom in this in a second. You'll notice these are surrounded by all of these simple cuboidal, that's what these are, simple cuboidal epithelia with a lumen, and there's kind of trashy stuff in the lumen. And those are all the PCTs and the DCTs, that is the proximal convoluted tubules and the distal convoluted tubules. So it would be possible, I'm not sure there, maybe one right here, a collecting duct could be there as well. So collecting ducts I can pick them out because they have very clean edges. They're nearly always nice rectangles, and the edge in the lumen is, you can see a nice edge, whereas these often look kind of blurry around the edge. We also bought these specially, these slides, to be stained so that there's a slight color difference between different parts of the tubules. So these brighter red ones like this are PCTs, and the less bright red ones, which I'm not seeing as often, maybe like these guys, are the DCTs. Now, because it's a struggle here, I'm not going to ask you which is PCT and DCT. You can just call all of these convoluted tubules. And they're really long, they're very folded around each other. And as you can tell, they are kind of randomized in here. There's no relationship between the capillary bed of one convoluted tubule that's allowing reabsorption to happen and the next convoluted tubule. It's all just a big tangle surrounded by lots of capillaries. So what about these capillaries and arterioles? Well, they left blood in this. So I don't know if you can see where I'm pointing are these little ghostly things. Those are red blood cells. So if we were to zoom up a bit, I know it loses some focus. You can see there are these semi-round things kind of shoved into here. That's a blood vessel. And that's going to be important more important than medulla, because I don't ask about blood vessels up here in the cortex. So looking more carefully at this particular glomerulus, you can see, maybe I'll use something other than a little, you can see with the arrow, these really flat cells that are simple squamous. These make they're tightly junctioned together, lots of tight junctions. They seal it so that the filtrate will stay in this and ultimately drain into the proximal convoluted tubule. The special stain also makes these really flat squamous cells look, I don't know what color you're going to pick, but grayish, sometimes grayish, greenish, and have very black, sharp edges. So again, that'll help you when we get in the medulla. So you can see another one here. Now the podocytes, I can't tell from the capillary endothelia. All these ghostly things in here, that's all blood that's in the capillary bed. But some of the nuclei, maybe the stain does distinguish, and I just don't know because there's some that are brighter red than others. So you just have to know that there's podocytes, which are the visceral layer of the two layers of this tubule, and the capillary underneath them. So let me see if I could move this down. So here are those ghostly blood. This is a large vessel. So this is either the afferent or the efferent coming in. Again, I wouldn't ask about blood vessels here, but good to point them out. See blood over here too. Sometimes it even looks red if there's enough of it. And then this doesn't look as idealized as the histology book, of course, but what you're searching for is you're looking for a row of cells just outside. This doesn't have really an indentation, but you can tell it's kind of disrupted right here. And they're in a nice kind of row. Sometimes, in this case, you don't see real well the other side, but sometimes you can see the simple cuboidal cells over here. So this is part of the DCT, or some books call it the very end of the ascending loop of Henle. But for a cortical neuron, it would be the DCT. So I usually call it DCT. We can't tell if this is cortical or it's a juxtamedullary nephron. They would look the same. So this row of cells are called the macula densa. And you'll say, wow, it looks like a mess here. Well, that's because there's all these other cells that are all crowded in here that aren't blood. And those guys are the mesangial cells that are on the outside as well. So the only way you can tell macula densa is it's in a nice little row. And we don't have a full one here. But in this one, you see at least some of the cells. Here's one cell, another cell, and there's a blob or something on top of that cell of the macula densa. And then you can actually see it's not circular, but there is a tubule out here with a space in the middle. So we'll talk about their functions more later. This is probably all the DCT continuing. It got cut kind of along its length here, and that's partly why this doesn't look like a nice circle, well, nice opening in the middle like this one did. Now, maybe here you see some mesangial. I'm not convinced I see cells in a row. So that one wouldn't show a great macula densa, but you do get the sense of a DCT sitting out here on the outside. It even looks kind of circular in this one. So this is the location they would be in, but they're not showing this nice kind of row of cells that are really linearly arranged. And they're always on the side next to the glomerulus. So I went through this slide, and let me stop sharing this somewhere. Hopefully it's still there. I have the labeled version. So this is the study guide. You guys seeing this now, the study guide?
Yes.
Okay, great. So it again enumerates what you should be drawing. I did add one phrase here because something is not visible. So we did talk about the ureter, the bladder last time, and kind of like in the respiratory, you're going to look for the four mucosal layers, meaning the surface epithelia, the submucosa, and then the muscularis externae. In the urinary system, the muscular externae have two layers. The inner one, let's see if I get this right, yeah, is circular layer, and the outer one is a longitudinal layer, meaning the fibers are going around the perimeter for the circular layer and along the outside for the longitudinal layer. And then you have generally adventitia on the outside with adipose in it. So for at least one of those, the darker stain, you really can see all those layers. And you can even see the lamina propria, which is part of the mucosal epithelial layer. So that one, you definitely can do all of the layers. The lighter one, I think you still can. It's just a little harder because the stain is so light on that one. The bladder, you can do it as well. The only problem is that the bladder is a big organ, and they didn't include the outermost layer. They cut it off, and depending on where the bladder is, sometimes it's cirrhosis, sometimes it's adventitia, but it's just not there. The last thing for the bladder, and you don't have to recognize it, but you have to know that you get to add a third layer of muscle that is obliquely, so you've got a circular layer, you've got a longitudinal layer, and now you've got an oblique layer. This is called the detrusor muscle because it helps collapse the bladder down to empty it. So the other two layers also help empty, but in the ureter, what they're doing is peristalsis and segmentation. They're moving it along, kind of like what happens in your GI tract. But by adding this third layer, you not only are moving things downward, you're also kind of collapsing the bag of the bladder. Okay, so I didn't embed reference slides for those. I think the histology book would be fine. You'd probably be able to. They're not very difficult slides, but if you have questions, of course, ask me about them. So with the kidney, you're going to do at least one drawing, minimally one drawing. You may find you want to do more just to make sure all of these structures are visible in the drawing. In the one I just showed you of the glomerulus and the macula densa, I pointed out all of these structures. The visceral layer is mixed up with the blood, the capillaries, but you could still know where it was. That's the podocytes that I talked about. Now, we haven't looked at the medulla, but we will. So to help you with it, because we just did this, first of all, are you still seeing this picture from the book now?
Yeah.
Yeah. Let me zoom in on. Oh, maybe I can't zoom in. Well, we can. This is what you are seeing in the slide. And it's all labeled. This is in the lab book. It's also in the textbook. You are seeing this thin parietal layer of the glomerular capsule, then that white space, which is where the filtrate accumulates. And you are seeing these mixed up capillaries, mesangial cells, and podocytes. And it was just a big bundle of cells. And then here's that very straight line, and they tend to be a little taller too, but sometimes that's hard to tell than the other cuboidal cells here. This is the macula densa, but it sits, as you can see, right next to this cluster of cells, which are the mesangial cells. And the book makes these all look a little more organized than probably they are in reality. These other purple cells in here are also called mesangial. So these are glomerular mesangial and extraglomerular mesangial. They have different roles, even though they have similar names. And then the afferent arterial and the efferent arterial. So this is a good reference picture that's a drawing for you to use with those glomeruli. And then I'm going to click on the labeled version. This is a different picture than the one you saw, but it's showing the same thing. There's a row of cells. This one's a little out of focus in the middle. Let me switch this over to a pointer again. Now I'm getting the pointer and the plus, I guess, because it's PowerPoint. You can't override PowerPoint, I guess. Anyway, maybe if I turn it into a pencil, it would stop doing that. Yeah. So this one is a little out of focus, but it's there. So you have at least 1, 2, 3, maybe another one here, 4 cells in a very linear way. And then out here, you see the rest of that distal convoluted tubule. and out of focus, but in here you can tell there's a bunch of cells, and even there's some over here. These are all the mesangial cells that are extraglomerular. This is just a big mess with blood cells. These are the ghostly things in here. And then the podocytes, the endothelia of the capillary, and mesangial cells all just bundled all together. And this would be a PCT. See how the edge is very irregular and kind of, well, it's blurry. It's not blurry because the picture is blurry, because see, the nucleus is in focus. It's blurry because the membrane's highly folded, so it's really difficult to focus on it. And you have some large blood vessels here with the ghostly blood inside of them. So, and here the DCT. Here's the brighter red PCT, the duller red DCT, still looking a little fuzzy in the interior side. Now, why they're fuzzy is they have microvilli, so lots of microvilli, which the collecting duct doesn't have. Okay, so let's stop sharing this. Well, maybe we'll go to the next one since I'm here. It's not going to let me do that until I get to the mouse. Okay. So again, this is for your reference labeled. You are going to need to draw more than one picture of the medulla. Now, why that is that even though they're all crammed together, it's sometimes hard to have a good view of both the descending loop of Henle, the ascending loop of Henle, and a collecting duct in the same, high magnification view. This one shows everything but the collecting duct. I don't have any collecting ducts on this picture, but you will. And collecting ducts are very distinct. So instead of, this is like, you can see the lining of the lumen, that these are kind of cuboidal cells. And this one has a kind of bright red color. The collecting duct doesn't. It's kind of a duller pink, lighter pink, more like what you're seeing over here. And the inner edge, you can almost always see a black line there because it's not got any microvilli. If it's well focused, you can see the edge of a collecting duct. Whereas here, it's almost so blurry, you can't even see the lumen very well because the microvilli are reaching in amongst the lumen of this tubule, and it all just looks pink and blurry. But this would be an ascending loop, a thick segment of the ascending loop of Henle. And this is a descending loop. And how do I know that? Let me go back to the better pointer. See these greenish, grayish, I don't know what color you want to pick. They're really super, super flat squamous cells that are lining the edge here. You can see them on both sides. And yet the middle either has, well, it doesn't have red blood cells. It either has nothing like you're seeing here, or sometimes you see kind of a trash in there. It just looks like little crumbs or something. If you're seeing squamous cells like this, and again, they're really super skinny, and you see blood like in here, there's a squamous cell here. It's really, really flat. And it just looks a little like a gray or darker line. You can see a little gray or darker line here. But there's blood in there. Those ghostly things are blood. So same thing with here. Here you can see that gray, greeny color of these squamous cells. Those are the vasa recta. They're the capillary bed. And they're filled with the blood to help you distinguish the thin segment of the descending loop of Henle from the vasa recta in these medulla. And you also get the impression of how squashed everything is together. And why you want that is you want rapid diffusion to occur. Let me get out of this thing. So I apologize. This next one is really out of focus, and I had this in the old study guide, so I've just copied it out of it. But it is showing you a collecting duct where the cells are really nice. You could actually, on this one, even though it's not in great focus, see all four sides of this collecting duct. So this is where the filtrate would go. This is another collecting duct. This is actually a descending loop, and all that darkness that's kind of out of focus is the squamous cells on that side. And this is an ascending loop with its brighter red color. So when you go looking for a collecting duct, they're pretty easy to tell. This one, the color, unfortunately, is not as good an indicator as it will be in your pictures, because these tend to be a light pink and not this bright pink at all. This looks pretty close, but they look a very much lighter color pink than the others. Okay, so just to finish that up, I'm going to stop sharing this, and I'll show you at least one of our medulla slides. Let me get back to the slides. And there's a couple versions of this one. At least I think I have a medulla. Oh, here it is. Of this medulla one. Let me get to share screen. I think there's just two slides. This one, you see lots of blood, right? So lots of vasa rectus here. And then here's something which just has kind of trash in it. Hopefully you're seeing this one again. That's probably a descending limb, because see that greenish, gray, black, whatever. That I might be one as well, because it looks squamousy. Here's a ascending loop, because it's that brighter color. I can't say it looks as quite as blurry, but it's that bright red color. And here is the light color and the much more, where you get to see all the edges. They don't look cuboidal right there, of a collecting duct, but you can kind of see the edges. The cells tend to be a little bigger too. Unfortunately, it has a little bit of that pink here too, so it's not coming out light. If I can, I'll get Kevin to take another picture of a collecting duct that's in focus for you. But if I were to do this, this would be ascending. Here is, where was it? This descending here with a wider lumen. And then vasa recta here, vasa recta all through here. So you need to, oh, here's another descending, you need to kind of get this search image down. So you don't have to label all of them, just so you have a search image, so that if I picked one of these and pointed at it, you could tell me which of the three things you are seeing in there: an ascending loop of Henle, descending loop of Henle, or four things: collecting duct or the vasa recta capillary. Okay, so I could look at if there's any other slide thing that's worth looking at. Maybe just one. This is the ureter, the darker one. Whoops, I have to grab you guys, not that. And you're seeing this one okay now? Because I kind of fumbled around. Okay, great, thanks, Kelsey. So here is the lining. Now notice it's stratified. What it actually is transitional epithelia. So whether you're in the bladder or the ureter, it's transitional, because transitional, if you go back to 103A, stretches and those cells change shape. Right now, they look kind of cuboidally round, whatever, but as urine goes by, they can stretch really wide and get flattened out. So over here, they can look a little more flat. But as they can change shape, they're very elastic and rebound back. So here is that transitional epithelial layer. And then I think this one shows it pretty well. You can see this is a different connective tissue in here than right on the base here, where you have a lot of blood vessels. It looks redder because there's an intense capillary bed right there. That's the lamina propria. This stuff is the submucosa. And then these elongated cells, these are smooth muscle. So I wouldn't point at one and say, is this longitudinal or circular? Just know that there is a longitudinal one and a circular one. And then finally out here, you have adventitia with there's a blood vessel, a lot of fat, and another big blood vessel, and just connective tissues out here. This is all the third layer, the adventitia layer. So the urine would flow through the middle here. So I think that's the idea. You're going to go through the others as well as this one and be able to label those layers, know their tissue subtypes, why they are the tissue subtype they are. Again, as in respiratory, this is a fibrous connective tissue. This is fibrous connective tissue with adipose in it. The biggest change is this one, which is transitional epithelia. And this is actually visceral smooth muscle instead of multi-unit smooth muscle that you had in the bronchi. Now, why you want it to be visceral is because when you stretch something passively and it's multi-unit, it would cause it to contract. And you don't want that because your bladder would be spasming all the time. You want, as the urine fills the bladder and starts stretching those smooth muscles, they reflexively, and I don't mean by neural pathways, it's just the way the muscle fiber is built, they will relax, kind of like the myogenic effect that we talked about, where they contract when they get passively stretched in blood vessels. We're going to come back to that myogenic response that we talked about in the cardiovascular system as well, talking about the arterioles that bring blood in to the kidney. So again, if you've forgotten to review the types of smooth muscle, it's going to come back because we're doing a lot of smooth muscle. So let's see, what time is it? Oh, I've talked for quite a while. If you guys have any questions or you want to be put in a group, you can let me know and I'll let you work on these. I don't want to go forward with any more lecture questions, but I'm happy to answer your questions because there's only 7 of you here. Everybody else couldn't make it back to the 1 o'clock. So just put it in chat or shout out if I'm wandering around on the computer doing other things. And then, as I said, if you have questions about the models or the slides, then, or how to label them, you saw that one slide in the study guide tells you how to label and what you need to label. And let me know. Okay, all right, so I'm going to just turn off the camera and the sound, but Hi, Kaylin. I tried to privately message you back that you could share the slide with me and I'd help you, but for some reason it says you're not here. I don't know why it's not letting me message you back privately. So if you want to share the slides, we could look at that.
I'll go find the link for it right now. Okay, I sent the link in the chat.
Okay. All right. What is going on? All right. I don't know why it's not letting me log in. Let me try it with. Ay, ay, ay. Ay. Let me share the slide and see. I don't know what's going on. It's giving me a hard time. It did this when I tried to log in from my phone, so maybe I screwed something up. Okay, respiratory slides. Oops, I gotta go back. This one, two. Histology, respiratory. So I'm gonna guess it's the one that says bronchi and bronchiole.
It's the one that says terminal bronchiole.
Terminal, okay. All right. I'm gonna make it a bit bigger. Share. I mean, I think this is blurry enough that I would have a hard time testing you on it, other than the knowledge that that happens. But let's see if I can get it to be a mouse and stay a mouse. Here we go. Can you see it now, Kaylin?
Yeah.
Okay. So hopefully this is clearly as clear as anything is that's out of focus like this.
Yeah.
These are tall cells, so they're going to be pseudostratified since we're in the bronchi. And then as you go into this, some of this is just disrupted, but you can kind of see that these cells are getting shorter in here. So they're tall here and they're shorter in here. And it's, as what I mean by disrupted, is the plane of section is kind of odd, where you're not just able to follow it in like the pictures in the lab book. It looks like it cut across, a bend or something. So you got this bridge of tissue here. So probably the bronchiole is going in here and underneath this bridge of tissue and underneath some of this disrupted tissue. It's almost like something dipped down and the knife just slivered right through it. So that air is probably ending up in here. And again, the lining here, you can see, are getting smaller and smaller. And then eventually they become the squamous cells that are along here. These might even be squamous at this point. But I grant you that both the contrast here and the focus is not making it so that you can clearly see this as pseudostratified. You just have to know that if it's tall, the only option here along a bronchi is pseudostratified. This is simple squamous. This is leading into it, and you're seeing kind of somewhat tall cells here that then are getting shorter and shorter. There's a picture, I think, in the textbook and the lab manual where they have a better terminal bronchiole, and you can actually trace it all the way out to the alveolar ducts, and you can see these getting kind of shorter in that picture. Does that help? See, there's a couple more. Oh. Is that okay? Are you there?
Yeah.
Okay.
Yeah, sorry, my internet connection's a bit unstable right now.
Okay. All right. So I'm going to stop sharing. All right. So, good. All right. Let me know if you have more questions. We'll try and find
I think I just had another question about the lining on the posterior and interior larynx too.
Everything in the larynx pictures we have is stratified squamous. You'd have to go a lot deeper.
Okay.
And I think there might have been, let me see if there was in the, if I close this, it'll probably show it. May only have put those two larynx. No, there's inferior larynx. There were two. There was a low magnification, I guess, of the inferior larynx. Yeah. And let me make this bigger and see what it's looking like. Yeah, I think it's all stratified squamous. I don't think I took a picture inferior enough in any of these to show you the pseudostratified. So I would just know that you have to get lower than, that if you get low enough in the larynx, you will come across pseudostratified, but I didn't take a picture where it's inferior enough. Is that okay?
Yeah. Thank you.
Everything's stratified squamous along both edges. So pretty much any edge you see, unless it's a cut edge, that is artifact. Okay, is that good?
Thank you.
Okay.
Yeah. For the cartilage, would it be a hyaline cartilage or elastic cartilage in there?
Hey. How about you?
Professor?
Yeah.
For the posterior larynx, would that be would it be a hyaline cartilage for the arytenoid?
Yes. All of the cartilages except the epiglottis and the larynx are hyaline.
Okay.
Okay. And what happened? Here it is. Okay. Oh, which I don't have, of course. I don't have. You want to eat any more of this because I'm going to throw it away otherwise. You want any more? Oh, I'm going to throw it away. Okay, guys, I have to switch over. I think that's right, isn't it? The times, I'm so confused these days. Yeah, I have to switch over to the office hour. So you're welcome to keep working, or if you have questions, find me in the office hour. But it starts in 6 minutes. So I'm going to end this one and see you on Thursday for the lab test at 9:20. Okay. Thanks for understanding today.